Recall that if your two mutations are on two different locations of your phage genome, so this is the first location, this is the second location, you will still have a functional copy of the gene and make functional protein, and the bacteria will die. That's the whole idea of the complementation test, right? However, what exactly do you think happened inside the bacteria when it got killed? You might think that, since the phage has the ability to kill now, some of the wild type phage that has no mutation was born. So like this is the wild type phage that is just clean, like there's no mutation on it. Well, that's not entirely true. The actual recombination of these two functional genes are pretty rare. Recombination just means, okay, here's your functional gene, here's another functional gene. Recombination means this is attached to this. So like they come together and produce a clean gene. That's the basic idea of recombination. If the other two mutation part recombined, so like this phage would just like straight up be dead. So that's why there's no phage with both mutations on it. Okay, so this is the rare wild type genotype. In your successfully complemented plate, it is just showing the wild type phenotype. That means there are two phage injecting their DNA into one bacteria. Their whatever toxic functional proteins made in the bacteria, like the functional protein made the bacteria die. So you can have two phage, both with some kind of mutation on them, and they can still work together to make the bacteria die. Okay, so remember, this is a two-phage work. If you have a phage with the wild type genotype, like this, so like only one phage is enough to make the bacteria die, right? So that is what we're trying to figure out by designing this experiment. So are there actually wild type phage being created through recombination? The tricky part here is to make sure only one phage is infecting one bacteria. So one phage for each bacteria. Because if you just have one phage with the mutation on it, it cannot make the bacteria die. But if you accidentally get two phage with different mutations to infect the bacteria, it can show the wild type phenotype, which is to make the bacteria die. And that's just our complementation test, right? So in this experiment you're going to design, you need to come up with a solution to ensure only one phage is infecting the bacteria. So when we see bacteria die, we know it's the actual wild type phage that's doing the work. By the way, if you're stressing about this post lab, please try not to scare yourself by thinking this post lab is super complicated. It is not actually not. Um, so once you understand everything and you're able to explain it with your own words, everything just kind of flow through, everything is coming together, and you'll feel much more comfortable doing it. And don't worry about the fancy terminologies. I don't really care if you're using the fancy words from the lab manual. As long as you're able to, as long as you understand it and you're able to explain it with your own words, that's good enough. The whole goal of this lab is to let you understand the concept, not to teach you how to write fancy lab reports.